This is the second webinar of the early career network of NuGO. And following the previous webinar that maybe some of you have watched, which when we were talking about Metabo Analyse, which is a tool for metabolomics data analysis. Today we have Olivia Alder. She's going to talk about MetaCore, which is a database and a tool for omics data analysis. So Olivia has had her PhD at the Imperial College London, and she did a postdoc working in cancer in the BC Cancer Agency in Vancouver. And then following that, she was working as a bioinformatician at the University of Leeds in the UK, always working with cancer and analysis of omics data. Now she is a solution scientist at Clarivate, and she works, or she is quite acquainted with MetaCore. So I think Olivia can start right now. I just wanted to make a couple of words for those of you who are not familiar with the GoTo platform. So don't worry, we cannot hear you. Your microphone is not active. So if somebody needs to talk or to make some noise in your office, feel free. You have a control panel in the right-hand side of your screen. There you can see that you have a space to type questions. So even during the presentation, feel free to type any question or comment that you want to be read at the end of the presentation. Olivia is going to speak for about 50 minutes, and at the end we are going to have a little discussion. So Olivia, thank you very much for accepting the invitation and for being here with us today. So feel free.
Okay, so you're welcome. Thank you for the invitation. So I just wanted to make it clear that you guys as NuGO consortium members have access to MetaCore, and we really encourage you to take advantage of it. This is what I hope to cover with you today. Basically, what is MetaCore, because I'm guessing some of the attendees are not so familiar with it. And then I was going to go into a live demo. We're going to use it for some data mining, and then we're also going to import our own data into it and analyze some results. Okay. So what is MetaCore? It is essentially a database of molecular interactions. Okay, so there's close to 2 million molecular interactions within our database. And where do we get these interactions from? So there's an editorial team, and it's their job to go through the literature, to read paper after paper and pull out those small-scale interactions based on small-scale experiments. So for example, if there's a co-IP in a paper and it's proven that two proteins interact with each other, they'll pull out that interaction and index it in our database. Okay, so they cover a wide variety of journals, and they also attend conferences to keep their eyes open for things in posters and during talks. Okay, and the editorial team has been doing this for over 10 years now, and it's made up of MDs and PhDs. And we really pride ourselves on the manual curation of our database. So we don't use text mining. It's really people reading the full articles and pulling out the information, so that when you want to find it, you can get straight to it. You don't have to go through reading all those different papers. Okay, so one thing we're quite well known for is our pathway maps. So these are the canonical signaling pathways, or disease associated signaling pathways that can be pulled out from this database of interactions. Okay, so then the more well-trodden paths of cell signaling. I like to think of this process as the editorial team are basically the flame. This is the wealth of information out in the public domain in the literature. The editorial team with a flame, and they're basically distilling the scientific findings so that you can get straight to the hardcore knowledge. Okay, so I would encourage you, instead of going to PubMed, you go into MetaCore and pull out the information more quickly. Okay, and of course you can read the papers in full in PubMed as well. So you can use MetaCore in two ways. You can use it as a data source, so you can go in and knowledge mine within it, or you can upload your own data, or even public data or competitors' data that they've just released in their new paper, and you could put it into MetaCore, and that will help you work through and analyze it and really put the results into context. So it really helps you look at the results in the landscape of what's already known, in the context of what's already known in the literature. Okay. You can look at various types of data within MetaCore, and you can also look at them at the same time. So here we're showing variant data being displayed at the same time as RNA-based data. Okay, but you can also upload metabolites, variants, and gene expression. Okay, or even structures like compound structures. Okay. So that's what MetaCore is. Now I'm going to jump into it live to show you how you can interact with it, how you can use it. Okay, just bear with me when I change screens. Okay, so once you've logged into MetaCore, this is the page that you'll see. Over here, this won't be populated quite so much because I've used it quite heavily, so I have a few data sets within here. But it's basically divided into 4 panels. All your tools are over here on the right, and a description of those tools are down below. And then this is where your uploaded data sets will live. Okay. So as I mentioned, you can use MetaCore in 2 ways. And the first thing we're going to do is data mine within MetaCore and see what's in there that might be useful to us. Okay. So you could search for your favorite gene. I'm going to use the example of BMA L1. You can use what we call the easy search up here on the right. Search. So MetaCore is based on rat, mouse, and human data. So beware of choosing the gene most appropriate. So I'm going to have a look at the human. So you'll notice here that we have all the different synonyms associated with the gene, and it's also called this. So we'll click through. And this takes us to what we call a gene homepage. So it's kind of like NCBI in that it'll have a description of the gene, its function. So we can see that what it is, it heterodimerizes with clock and binds to EBOX enhancer elements and regulates these genes. Okay, and that these genes are important. Defects in this gene have been linked to infertility, glucogenesis, and lipogenesis. So just give you a summary, quick summary, what this gene is known for, what it interacts with a little bit, and the diseases it's associated with. Okay, but where it kind of differentiates itself from NCBI is over here on the left, the table of contents. I really, really encourage you to dive into this. Okay, so it's got the diseases that this gene is associated with. So we can see the diseases that it's associated with, but you can also see the interactions. So as I said before, we have a huge database of interactions within MetaCore. Okay, so here we can see that this gene has been shown to have some interactions with about 370 different molecules. We can filter these interactions if we wanted. So say we wanted to know what affects this gene. So what's upstream of this gene, we can just filter based on that. We could also filter based on the mechanisms. So if we want to know that the interaction is by direct binding rather than microRNA binding or ubiquitination. So we can click binding and click apply. And then we can see that genes, as expected, the CRIs and the PERs are shown to interact with this gene. Personally, I didn't know that part 1 was meant to interact with this gene. So I can say, oh, I'm not sure about that one. Where's the evidence for that? And the evidence will always be on the right over here. So if I click through, then we can get straight to the paper in which this evidence has come from. So we can see it's from Cell 2010. We can link through by the PubMed ID, and we can also see how the editors have indexed this interaction. So we can see the sentence that they pulled out from the paper, and what I find really useful is that you can see the tissue in which this interaction has been proven and the methodology that has been used to prove it. Okay. You might have a favorite gene that you hope interacts with this one, and you have some kind of inclination that another gene is influencing this gene in some way. For example, my favorite gene is YAP1, and I couldn't find that it interacts at all with this gene here. But I feel like they are connected in some way. So MetaCore has some really cool network building algorithms, and I would encourage you to take advantage of these too. Okay, so you can click here on the build network, and I'm going to show you how you can connect the dots using our database. So one of the algorithms we have is called shortest path, and what it enables you to do is to select two genes, or microRNAs, genes, compound genes, two molecules. I'm just adding YAP1 here. So I have an inkling that YAP1 is somehow connected to BMAL1, okay? And I think it's connected quite closely. If I thought they weren't connected a bit more loosely, then I could add an additional number of steps between them. And I can click Build Network. So although I saw by looking at the table of interactions that YAP1 didn't directly interact with this gene, I'm now asking it, saying, are there any in-between molecules that interact with the two of them? Okay, is there a connection between them in any way? I'll just wait for this network to load. Hopefully it won't take too long. Okay, so these are the genes that I started with, BMAL1 and YAP1. And then these are all the ones that connect them. Okay? So we can see that there are a number of ways in which, although these genes weren't directly connected, only by adding an extra step in, there can be a number of methods in which signaling pathways or signals could pass between these two genes and have an effect on one another. Okay, we can ask this to show us the mechanisms by which these interactions take place. So we've got binding here, this is phosphorylation, this is transcriptional regulation, and you can always link back to the literature that those interactions were based on. So the networks are interactive, so I just clicked on the link, and then here's all the citations that support that interaction and the tissues in which that interaction has been established. Okay. So, also, if you wanted to share this information with your supervisor or another member of your team, and you can always do that. So you can export the image of this, saying, look, I was right, there is some kind of connection between these two genes. Look how many different pathways the signal could go through. So you could just export the image, or you could export the interactions themselves. So if we click interactions down here, and test, we select the columns, you can see that it's going to pull out the two different objects that are linked together, the effect and the mechanism. So whether it's activating, the interaction should be activating or repressing, and also all the references that are associated with that interaction will come out. Okay, click export. So we're really delving into the database and trying to use it to our advantage. So this is the kind of information that you can pull out and share with colleagues or supervisors, etc. And you can say, or you could prioritize perhaps which pathway you're going to follow up, because we can instantly see that this this pathway between the two molecules has more citations supporting it than, for example, this one. So you might want to prioritize the experiments by looking into that interaction first, for example. Okay. Okay, so we used Easy Search to search for a gene. You might not be as gene-centric as me. You might not have a favorite gene that you want to type in and look up and find everything you can about. You might want to type up in a disease name instead and find out everything. Oh, that did not quite go to plan. Diabetes. Okay, search. So you can also search on a disease name or even a compound that you might be interested in. So you see here, first up, it will bring genes to the top, but if you look at the objects found, it's categorized the results. So I could click down to the diseases, and then disease diabetes mellitus is at the top, and then you have all the more precise definitions below. So we click through just to show you what a disease homepage looks like. So it's very simple here, but again, all the really good stuff is in the table of the contents on the left here. Okay, so we can have a look at all the genes associated with diabetes by clicking on the left. It takes a little bit of time as there are quite a number of genes associated with it. Okay, so we have a huge table here. Over 7,000 associations have been made with the diabetes, but you can see that many of these are at the DNA level, so a SNP haplotype. So if we're not interested in those, maybe we want just protein level kind of associations, we can of course filter this table down by using the options on the left. So perhaps we just want protein level, and perhaps we wanted to just limit the associations from ones that from a certain tissue. So I might just want to restrict it to adipose tissue. I click apply, and then I've got straight down to 12 targets that I might be more interested in paying more close attention to and following up with. Okay, so we searched on a gene, we've searched on a disease, and we filtered it. I also wanted to let you know that hiding over on the right here is an advanced search option. So if you really wanted to get down to some really quite tricky questions, for example, you just want to pull out all the genes that have an association with BMAL1 and an association with diabetes, for example, then you could use the advanced search over here on the right. I'll leave that for today because it's meant to be more of a tips and tricks session. I just wanted you to be aware that you can perform some quite powerful searches on the database using the advanced search. Okay, so I'm going to head back to the start page. Close a few windows here. And let you know just one more way that you can delve into the database without having to upload any of your own data. So over here on the right is a search and browse button, and here you can just really have a look around what we've got in the database. I'd encourage you to look at the pathway maps here. I've opened them up already because there's so many of them it takes a little bit of time to load. So this is the page that you'll come through to. You can see that we have what we call canonical signaling kind of pathways. And we have a group of folders dedicated to stem cells, and then also what we call our disease maps down the bottom here. So there's a number of different maps within these folders, but I thought what might be most relevant to this group is pointing out that we have a dedicated folder for metabolic maps here. Okay, so and within each of these folders, they expand to more maps. So let's have a quick look at one of them so we can, if you've never looked at one of them before, we can get orientated as to what I'm talking about. So this is an example of one of our maps. And just like the networks we were talking about, they're interactive. So you can get straight back to the literature that supports the interactions within the map. But unlike the networks, these are meant to represent what we call more canonical signaling pathways or more well-trodden biochemical or molecular biology kind of routes through the cells and signaling. So the maps are restricted because there's only certain amount of interactions you want to put on one map, but we understand that all the processes in the cell are connected in some way or another to each other, and so we connect the maps to each other as well. So we can see that this is feeding into this pathway, and that will lead to this process. So you can click through and have a look at that as well. Okay. And then again, we can see from here that leads to yet more processes that you can click through to and have a look at. So you're not limited to just looking at these pathways. You can click on them, and you can also export them. So I'm going to show you one tip in that if you're interested in a particular process, a particular pathway map, you can export everything that's on it, just as we did with the interactions from the network. And also, I'm not sure how interested many of you will be, but you can get biomarker information out. So we have another database called Integrity, and it has a huge biomarker component to it. By exporting this, let's just do a test so that you can see. Test export. By exporting all the components on this map, it will show you which ones are being used as a biomarker in and in which diseases they're being used as a biomarker. Okay, so for example here, this gene here is being used as a biomarker. This one here in Australia, it sent me through to our other database. I didn't mean to do that. Test export. So for predicting treatment efficacy in ulcerative colitis, for example. So biomarkers are well characterized and can often have chemical assays associated with them. So it may be another way in which you want to prioritize your research, because what I found in research is there's always so many questions that can be asked. It's finding which one you want to focus on or which lead you want to follow up on that can be the trickiest. And the more evidence there is out there, that's probably the best lead to follow up, isn't it? So I just wanted to let you know that that's available. No, I don't. Okay, so that was really what I wanted to cover in terms of data mining in MetaCore. So we had a look at to see what interacts with the gene of interest. We built a network, and we had a look at 2 different molecules and built a network between them. And I showed you a couple of tips of exporting information out of MetaCore and how you could browse through the maps to find signaling pathways that might be useful to you. Okay, so now I'm going to move on to importing your own data into MetaCore. And your earlier careers network group organizers gave me the challenge of using a relevant data set that, well, a data set that might be more relevant to you guys as an audience. So I did a little bit of browsing and digging, and it soon became clear that nutrition and health and well-being and sleep are intimately linked. So I've taken the liberty of pulling a couple of data sets together that are based around that. So I pulled some metabolite data from this journal here, from this journal article here, and I've pulled some microarray data from this article here. So both are liver-based and are based on the finding that BMAL1 disruption has a link with high-fat diet and the disruption to the metabolites and the circadian rhythm. Okay, so as I've pulled the metabolite data and the RNA-seq data from different data sets, please don't expect any exciting scientific revelations. And if you can point me towards a data set that has the two coming from the one source, I would really appreciate it for future webinars. Okay, so let's jump back into the software, and I'm going to show you how I upload the data in. Just close a few windows here. Okay, so as I said, all your tools are over here on the right on your start page. You click upload here, upload experiments. And this is a wizard to help you upload your data. So it tells you how we expect your data to appear. So a gene identifier of some type, an expression value followed by a p-value is the ideal scenario. We can use a different identifiers, whichever your data is in. And I'll just show you if I choose file here. Next. Could one of the organizers confirm that you're able to see my screen still? I just got a little window pop up saying I was no longer presenter.
Yes, we can see it. Yes, fine.
Perfect. Okay. So I've uploaded the data, and the wizard has tried to predict what data I have in each of the columns. So I have my gene symbols with aliases on the left here, and then I have the fold change between, I took some data from high fat versus normal diet at I think it was 20 hours CZT, and then I took, that's the fold change and the p-value. And click next. There's mouse data. Click next. Okay, so once it's uploaded, the bar will fill up. You can go to show location. Okay, so now we can see that this has appeared down here with a green tick next to it. So active data sets will have a green tick next to them. If you wanted to inactivate it, you could just click it and it would go from green to grey and no longer be active. Okay, so once you've got your data uploaded into MetaCore, we highly recommend that you have a look at it. So we have a couple of different ways in which you can do this. So if you go to view and then signal distribution, we can see that the majority of this data is falling between 0, 3-fold upregulated and 0 and 3-fold downregulated. And that's not really useful when we're wanting to set thresholds that we know that majority is in between here. So you can click on that and you can get a higher resolution. So you see the axis has broken it down more between the 1 and the 3. And so we might perhaps want to put the cutoff, probably lose this bit, put the cutoff somewhere around here. Okay, so it gives you an idea of the distribution of your data and where you might want to set your thresholds. So once you've decided where you'd want to set your thresholds, you could filter experiments. And type in the values that you want to filter by and click apply. So if you were unsure about where you wanted to put your thresholds, you could do this a number of different times and then compare the results. So you could be less stringent and more stringent and compare the outcome if you wanted. And I don't know what I just did there. Let's have a look. Let's go to the start page. Click refresh. And then some filtered data should populate. That was the curse of the live demo there. Okay, I was just muted there for a second. Is it okay to continue? Yep. I'm going to take that as a yes. Okay, so we have our filtered data set now there, and the original is also untouched here. Okay, so by filtering, you can always go back to the original and apply different filters if you should wish. One thing that's very important to do if you're wanting to do gene ontology enrichment analysis, so basically see if your differentially expressed genes are involved in a process more than you would expect by chance, you must set the appropriate background list. Okay, and to do that, you can go to Tools, Set Threshold and Background. And for this experiment, because it was a microarray, we can just choose from the drop-down list. There was a mouse. Okay, so once you've applied your filters and your background list, then you can go and do an enrichment analysis. So we have a button here called Workflows and Reports. I recommend doing the enrichment analysis in here. So click Enrichment Analysis. Make sure you've got the appropriate data set selected. Click Next. And then if you wanted to, you could apply filters in here, but because we've already done it, we'll just click Apply. And what it will do is start to do enrichment analysis on all these different sections of the database. So we have our Pathway Maps folder, Process Networks, Diseases, Toxicity Networks, and Metabolic Networks. Okay. So it didn't surprise me to see that this methionine pathways and transport was enriched, and because the authors had also seen this in the publication. And so to have a look at how enriched it is, like what members of the pathway are enriched, we can click through. Okay, so it's a bit daunting to look at first, but I'll just talk you through a couple of things. So you can see that we have different symbols here, and these represent different types of molecules. So kinases, transporters, receptors, this kind of thing. But you can always have a look at the legend by clicking there, and it'll give you a legend you can print off. And you'll notice as well that we've got some red circles or blue circles by some of the members in this network. So this symbolizes the fact that these have been down-regulated by blue or up-regulated in red in our data set. Okay. So we are having a look at RNA expression at the moment, but I also mentioned that I took a metabolite data set. And so you upload this in a similar way to the RNA data set. Click Upload Metabolites, and it will give you the upload wizard. You can use a number of different identifiers. I just used chemical name and then selected the file. So it's here. I'm just going to activate it now. I'm going to activate it here. That's the start page that I'm currently on. Close that one. Now, when I refresh this, we should be able to see the metabolite data is also displayed. So now we can see the metabolites represented by the purple hexagons. Also displays some symbols next to it representing the abundance of these metabolites in the data set, or not. We can see that if we click here and right click and select downstream, this will circle everything that's downstream of that methionine. And we can see that these are down-regulated over here. And also these over here are down-regulated too. But if I'm perfectly honest with you, I find this a bit tricky to visualize. And I wanted to show you some tips and tricks how you can get a better visualization of it. So if you go to the enrichment analysis window that we originally saw that this methionine pathway or network was enriched in, and if you click over here and click that 5, this will show us the members of the pathway or network that were enriched. And we can say, let's go build a network from these and see what they influence. So if I say expand by 1, and I want to know what's downstream of these components of this pathway, what knock-on effects is it going to have? So you can click downstream and build. Okay, so then we can see what components are being upregulated and what metabolites are being changed. And we can see that in a much clearer way without all that noise of unaffected molecules being involved. So it gives us a much clearer picture of our data and the process that are being affected. Okay, and of course you can, just as we did before, you could export the image, or you can just save it as a network to come back to at a later date, or you could export all the interactions and the supporting evidence just as we did before. And you can also put the effects and mechanisms on top. Okay, you can make Okay. So that was one way I like to simplify the visualization by really taking advantage of those build network algorithms at the top there. One other thing you could do is you're not limited to the enrichment analysis. Although it's interesting to look at the enrichment analysis, we should probably take a minute to do that. So we saw a number of pathway maps were enriched in liver regulated kind of processes, and we can click through to those. But given that we know that the methionine has previously been reported as a significantly upregulated metabolic process, we might want to have a look at our data on all the associated maps associated with methionine. Okay, so we can just search methionine just as we did with the disease and the gene earlier on, and we can go straight to the pathways. You can see the pathways, gone straight past them. And go to the maps that are associated with methionine and view our data on those maps. So you can see the components that are changed. Instead of designated by a circle, they're designated by a thermometer, and you can hover over them and see the data. Okay. So I've talked about what MetaCore is. I've shown you a couple of ways in which you can delve into the database and explore your gene of interest, your disease of interest, and extract information out of it to share with your supervisors and colleagues. We had a look at uploading our experimental results and had a look at finding enriched pathways and networks and how best to visualize those. So we've got just coming up for 10 minutes for questions. So I want to hand over
Unmuted.
Take a pause for a minute and have a look at the questions, if any have come in.
Yeah, thank you very much, Olivia. This was really nice presentation, and you can see how powerful and useful the tool is. I particularly didn't know it, but both as a database and as a tool for data analysis, it seems to be pretty good. We have two questions from Yogatama. I can read them for you if you want. So the first,
yeah, that would be great.
For enrichment analysis, how to do it if I have Illumina data, since you showed before choosing from Affymetrix, etc. Was it clear?
Okay. So you've got RNA-seq data. Is that my understanding?
I don't know. Yogatama can bead array data.
Bead array data. Okay. So you can also let's have a look at the background list tools. Set background list. So you can also set it to like a customized gene list as well. So as long as you know every gene that was on your array that you've measured in your array, then you can upload a gene list and use that as your background. So this, because we're not limited to RNA data, so there is arrays, but we're not limited to that. For example, if you do a metabolomics experiment, you can't then use an array as a background list either. So we have the ability to upload a gene list and use that as your background list. Okay, so as long as you've got your list of identifiers that were measured in your experiment, you can use that as your background list. I hope that answered your question.
Yeah, there is another question. How to get to know whether the pathways is upregulated or downregulated? Any measurable score? But maybe that's the thermometer that you were mentioning?
Yeah, so I think you're trying to kind of, because obviously there can be an upstream molecule that's upregulated, but further down in the pathway something might inhibit that or turn it off or be down-regulated, and so the cascade wouldn't come all the way through to the bottom. So you can, when you do your enrichment analysis, you can select just to look at down-regulated genes or just to look at up-regulated genes, but there's no way of, there's no score associated with you know, the outcome of your down-regulation or up-regulation, if that makes sense.
Yes.
Let's
There is one more from Guy. Is MetaCore as good for metabolomics data analysis as it is for transcriptomics analysis?
I have to admit, I didn't do research in that area, so I would really ask you to go in and use it and really give us some feedback. I'm going to be really honest with you, like the majority of our customers do use it for transcriptomics analysis. Lots of them just use it as a database to get information out of as well. But yeah, I don't know, to be honest. But you can be our test cases, and I'd love you to give us feedback if you're finding it useful or not, or if there's pathways that you think are missing. Please do let us know, and I'd encourage you to go and browse through those pathways and the metabolic maps that I pointed out to you, and see if it's comprehensive.
Okay, thank you. There is one more from Diu Vertke, I think. Sorry for the pronunciation. He's thanking you for the presentation, and he wants to know if you know how MetaCore differs from Ingenuity pathway analysis.
Okay, so they are one of our key competitors in the commercial space, and we pride ourselves on the database being manually curated and based on small-scale experimental evidence. Okay, so we won't just take, for example, genome-wide data. Say it's like a yeast-2 hybrid, and there's been an association made in a yeast-2 hybrid. We won't just take that and put it into MetaCore. It has to also be supported by a validation experiment, a small-scale experiment. I don't think IPA has those kind of restrictions in place.
Okay, one more, very long, from Guy again. When the number of genes that is uploaded for pathway analysis is increased in a data set, the p-value is improving even for random selected genes. Does the system correct appropriately?
That depends if you're selecting the appropriate background list or not. If you're leaving it as default, we highly recommend you to select the appropriate background.
Okay. One more from Yogatama. The top 10 pathways you showed, does it have a measurable score, such as its score that usually are got from PathVisio?
Yeah, so sorry, I didn't spend much time on that, but let's try and grab it up. Just need to get rid of the controls here. So over here on the right, you've got your p-value and your multiple tested correction for FDR. So it's based on the hypergeometric mean test, as we based all our enrichment analysis on. Okay. So we're just showing the top 10, but of course you can go further down.
Okay, thank you. I have a short question myself. So when you updated or when you uploaded metabolomics data, you can just upload a condition, right? Or you can upload several conditions. For example, for a given metabolite, more than one condition.
Yeah. So you can definitely do that with transcriptome data. When I was fiddling about with it yesterday, it seemed that because I had some time point data, you know, like 0, 2, 4, 6, 8 hours kind of data, and you would have to upload them as separate experiments. Okay. So you can upload up to 8 experiments at once, but you can't do it in the same file. So if we go to upload, yeah. So here you can do experiment 1, experiment 2, experiment 3, okay. and it will just automatically process them, but in the metabolite section, you can only do like one at a time. So I did IDs, data, and then when I was looking to see whether I'd do 2, 4, 6, 8 hours, I was uploading again, then 2 hours, then 4 hours, then 6 hours, and you can display up to 8 at a time. So unfortunately, you can't upload the whole 8 together, though.
Okay. Well, thank you. I don't know if there is any question that I'm not seeing, or if you have something else to say. Maybe Sarah, do you see some other questions or something that you?
I just read. It's not a question. It's just the last comment of Guy Verger that say that well, sorry for the follow-up on my previous question, and he said that even if the background is selected appropriately, his experience is that it is best to compare data sets with similar numbers of genes. I don't know, Olivia, if you want to just add something more.
I don't have anything more to add, really. I guess so gene ontology analysis is just that. It's one way of analyzing your data. I wouldn't put all of my eggs in one basket based on gene ontology analysis. It's meant to drive hypotheses and follow-up experiments and to complement wet lab kind of experiments, right? Like you wouldn't go into the lab and just do one experiment and then that would be the basis of your conclusions or discussions in your manuscript. Like you need to follow up from different angles, right? So I hold my hands up and I say I think gene ontology has its weaknesses, but I also think it can guide you and find clues in your data.
Okay, Guy, thanks you for the answer, and the last one by Martina Poletti that asks you if there is a way to overlap two canonical pathways or networks.
Yes, you can merge. Yes, you can merge networks. So although we can't merge pathways, what you could do, I showed you how to export components of a pathway. So you could export all those components of a pathway, make them into a network, and export all the components of a second pathway and make them into a network, and then you could merge networks. Do I have any networks built that I could show you quickly? Okay, I can't show you right now, but I will follow up with screenshots of how you can merge networks, if that's okay.
Okay, so I think no more questions. Thanks a lot, Olivia, because it was a very interesting presentation and several questions, and it means that also the attendees were really interested about the topic. And Darley, I think you have to give more information about.
Yes, I have just a last reminder to all the attendees that are associated with NuGO. We do have a how do you call that, a license to use the software. So you just have to contact our secretary, Ingeborg, and to know how we can use this. So the tool is there. It's up to us now that we have the training to use it. I would like once more to thank Olivia for the nice presentation, for her time, and to advertise the next talk that we are going to have in our webinar series. The date is not yet fixed, but we believe somewhere in June, Valentini Constantinidou is going to talk about career options, so kind of a career advice session, talking about career options outside the academia. She has her own experience as an entrepreneur, so it's going to be also a very nice topic. Do you have something else to add, Olivia?
No, just thank everyone for their time, and if they have any questions when they get to using MetaCore, feel free to contact me. I'll send on, you have my details, so.
Okay. Yeah. Well, this presentation is going to be available in the website of NuGO. It was recorded, so everybody can watch it again if you want and get more questions. So thank you very much, everyone, and I hope to see you or hear you in the next session. Bye-bye.
Bye. Thank you.
Bye. Thank you.